Hello everyone and welcome to this class. In this class we are going to learn about apoptosis and it comes under cell death mechanism. Okay. So our body is made up of cells, cells which form tissues and tissue will organize themselves into tissue level of organization which completely forms our body. And for that formation we all know that we need growth. But do you consider the term death which is related to the death of cell? No, most of us don't consider it. But the production of cell works in equilibrium with the death of cell. And this cell death can be classified into three classes. First is programmed cell death, which you can say is a sequential step so that our cells die in an pre-planned way. Okay. And second comes the accidental cell death, which we call as necrosis. Okay. This accidental cell death happens due to some shock or some damage, some cleavage, etc. Okay. And third is self-demise, which is autophagy, when the cells of our body eats up other cells. This is called autophagy. And what our main focus here is this apoptosis. Okay. The apoptosis is what we called as programmed cell death. The cells dying from apoptosis undergo characteristic morphological changes. And what are these morphological changes? Let us suppose this is a cell. First of all, this cell will shrink. Okay, so the walls will collapse toward inside. Okay, they start collapse. Then this it will condense. Cytoskeleton system will also collapse. As you know, as the cell collapse, cytoskeleton will collapse, then the whole cell will collapse. Okay, the nuclear envelope dissolves and nuclear chromatin condenses. After the condensation of the nuclear chromatin, fragment formation is there and cell often bulge outwards. Okay, you see, first it tried to shrink, then it bulged outwards. And there is a special point to note that there is sometimes the apoptotic bodies formation which also happens in this, right? Like if the cell is too large, it will first break down into smaller cells. Okay. And if the cell is large, then it breaks into membrane enclosed fragments called apoptotic bodies, right? And apoptotic bodies are chemically altered bodies so that the neighboring cells, which are macrophages and some other phagocytic cells, can engulf them. Importance importance of apoptosis is that it maintains the cellular and tissue homeostasis. And homeostasis, as you know, is the, here I have written it also, like self-regulating process by which living organism can maintain internal stability while adjusted to the changes in external conditions. Okay. And second is it provide defense mechanism, right? Like apoptosis is a kind of defense mechanism. Let us suppose there is a cell which is infected by a virus. Okay, so apoptosis will kill this cell along with the virus, so it can provide a defense mechanism also. And the third point is the development of immune system, right? So the same thing, it helps in immunity also. And the importance, it has some importance in embryonic development also. Like when embryo is developed, it is a cell, right? The cleavage which happens is a part of apoptosis. And as you know, we have our hands also, right? Sorry again for the rough drawing. Like this whole hand was at a time was a sheet of, you can say, skin, right? But apoptosis particularly happened, and apoptosis happened at these area. So what it did is, this separated our fingers from one another, and the result was our hand. Okay. Then is the regulation of neuron membrane is also happens by the help of apoptosis. The regulation of cellular viability by hormone and growth factor. Okay. That also need apoptosis. And the seventh point is defense against cancer. Okay, like cancer, uncontrolled, un sorry, they continuously differentiate and form a huge mass inside our body. Cancer, you all know. So it is a defense against cancer also. And cells that undergo apoptosis are harmful cells or cancer cells, and developmentally defective cell. If body have defective cells, then apoptosis will destroy those cells. Okay. Then excess cells are also destroyed by apoptosis. Unnecessary cells, obsolete cells, these are outdated cells. Okay, outdated means they are of no use to us. Then virus infected cells are also undergo apoptosis. And in animals, if you take the example of tadpole which develop into frog through metamorphosis, the apoptosis role is the tail removal. See, this tail is removed by the help of apoptosis, and this tadpole will grow and turn into a frog. Okay. Second, the apoptosis role is in finger separation. I have described you earlier like this. The fingers will separate and ultimately it will form a hand. Okay. See, rest of the things will be apoptotically cleaved. And apoptosis kills B and T lymphocytes that are either fail to produce either useful antibodies or producing self-reacting receptors against body. Right? So basically, when B and T cells, they are used for defense, memory, cytotoxicity, etc. So when they fail to produce useful antibody against antigen, like this is our antigen, and if they produce an antibody which is not attaching to the antigen, then these B and T cells will be cleaved. This is the first case. Or if the B and T cells somehow produce antibodies that work against body, we don't want good cells to get destroyed. That's why apoptosis comes into play, and it helps these B and T cells along with the antibodies elimination. Okay, and it eliminates lymphocytes activated through infection. Okay, this is also a good point. Then comes the activation of apoptosis. The apoptosis is triggered by protease, and these proteases are called caspases. Let me take the highlighter first. These are caspases, where C stands for cytosine and ASP stands for aspartic acid. And these proteases have cysteine, I'm sorry, not cytosine, cysteine, in their active site and cleaves their target protein at a specific aspartic acid. Thus, they are called caspases. Okay, there are two major classes of apoptotic caspases. The first is initiator caspases, second is executioner caspases. These executioner caspases are actually responsible for apoptotic process, and these initiator caspases, what they do is they activate this executioner caspases. And I think I have written it here also. See, this ICAS here stands for initiator caspases, so I have written it like inactive ICAS, which is initiator caspases, which are in the form of monomer. When they get the apoptotic signal, they activate and become dimer. Okay, and they activate executioner caspases, so they will work here. Okay, and executioner caspases normally exist as inactive dimers when it is cleaved by initiator caspases. Okay, like let me draw it for you. Like suppose this box is executioner caspase. Okay, so when initiator caspases comes, it will cleave a certain part of this caspase, and when it is removed, okay, when this is removed, this executioner caspase will get activated. Okay, and when this gets activated, then only it will perform apoptosis. Okay, and here is another important point, that first initiator caspases will activate many executioner caspases, thus increasing proteolytic effect by amplification. Okay, see, one initiator caspase will activate many executioner cascades. That's why is you can say that we need a small amount of initiator cascades to be large. Okay. To activate large amount of executioner cascades. Thus, I have written this word amplification here. Okay. There are two pathways for apoptosis. The first is intrinsic pathway and second is extrinsic pathways. Now, as in this video, I'm going to teach you about the basics of apoptosis. That's why I'm ending it here. But in the next chapter, we are going to learn about these two pathways of apoptosis. That's it for this class. Thank you very much and see you soon.